Dude, I feel bad because I was like real nervous because she's the only one here and I went
You're
too
fine.
fast.
I'm sorry, no, you're fine. I'm recording already.
Oh, it's red recording?
Yeah.
Cool. Alright, so we're gonna go sliding filament theory first.
Okay.
This is very simple. Alright? So I'm just gonna go with the pointer here on the thing. You can follow along on your iPad. But basically you have this neuron right here, your lower motor neuron. This one will release ACH, and this ACH will bind to the nicotinic receptors, allowing potassium to go in, causing a depolarization to occur, right? You also don't forget, she might do it, she seems like the person. You have something called acetylcholine transferase in the middle, which dissolves ACH, cool?
It's the enzyme that's
She could also ask you the kiss and touch theory, which is basically, remember like the vesicles, like kiss, or like they touch the end of the motor neuron, release the chemicals, and like they dip out. She can ask you that, just like remember there's the general stuff, cool? So anyways, you have sodium rushing in, causing depolarization to occur, and then you have the signal being sent. Now, during this time, you can actually have summation of the signal. So basically, I can keep sending the signal as long as sodium's rushing in and depolarization occurs. Just like basically when I'm having neuron-to-neuron connection, you can have temporal summation. This one you can have a muscular summation. Cool? It's an electrical current. You have to understand that it's electrical. Smooth muscle is different. Smooth muscle, we'll see later on, it can actually be affected by chemical and electrical signals. But for the muscle right now, it's just straight up electrical signals. Cool? So here are the steps. What I wrote, like I made mine somewhat summarized from hers. So the first one is just a voluntary contraction that's initiated by your brain. So think about it like this. Your brain is telling your arm right now to write something, right? It's not like it's like magically doing it. You're just doing it. Or sometimes like, for example, an involuntary form of muscle, if you just want an example of it, would be like sometimes when your eye just randomly twitches. That would be an involuntary form. But you closing an eye is voluntary. You blinking without you paying attention, that's involuntary. Cool? They're all electrical currents, and they travel from the upper motor neuron to the lower motor neuron to the motor end plate, and then releases ACH, which binds to blah, 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 blah. Then the action potential will pass down through here, and then it goes through the T tubules, or the transverse tubules that I told you about earlier, and that will release, or send the signal to release calcium, right? And this is just telling you, I like summarize those, like that 4, 5, and 6 into like that one step. You good, Chief?
Yeah, I just keep going.
Okay. So, when the potential will actually reach the T tubule, so this is me, here's the sarcolemma, which is basically the cell, releases this potential, electrical current, it'll keep driving. So you have to understand that it's like the main thing I want to nail right here is like its summation. Because I could send you a signal and it starts right here, but if I don't send any other one, it'll end right there, right? It's not going to keep going. So I want you like summation to send it. Like I want to keep like the signal strong. So you send the signal and it goes down through the T tubule. And all the T tubules are interlinked, like I showed you before. So you'll have them all doing this at the same time. It's not like you're going to have like a single muscle and a single thing just activated while the rest are like, I'm out. Anyways, the calcium will hit here and it'll hit your DHP receptor. As soon as it binds to this, it'll send a signal inside the cell to your RYR receptor. Cool? Or your rhodamine receptor. That's like its actual name. Ranodine receptor. Anyways, so the sarcolemma, right here, sarcoplasmic reticulum, that's what I told you, SR, will actually have stored inside of it a buttload of calcium. What happens is the RYR will get the signal and we'll just shoot a bunch of this calcium out. Now this right here into the sarcolemma, but right here is a passive diffusion. You will actually, since there's so much calcium in the sarcolemma, later on when we want to put it back into the sarcoplasmic reticulum, there's so much calcium in the sarcoplasmic reticulum that when we want to end a signal and we want to put calcium back in, we're going to actually have to use active diffusion, push it all back in, due to like the concentration differences.
Mm-hmm. It's all concentration gradients.
Mm-hmm. So do you remember when I talked to you about troponin? That's where all the calcium comes from and hits up troponin I, or troponin C, which then causes the conformational changes in the thin filament of actin, which go from C to I to T, then move tropomyosin, making the actin or the G molecule of actin available, allowing you to have a myosin head attached to it. Cool? So here's just a more detailed photo. Basically right here, you have your ACH release, does the signal is sent, and from right up to here is just your basic action potential, right? DHP or DHP, DHP. I don't know why I'm thinking DHAP because she screwed me up in class.
Honestly.
DHP, and then your RYR, and then it will release it, which will bind to troponin, etc. And this is what I was telling you in the first lecture when I told you those little yellow parts on the actin, that's your globular actin, that's what the tropomyosin or the myosin will actually bind to, and what the tropomyosin is actually covering. Cool? So it actually shows right here. You have them blocked, and then calcium comes in, and now you have the yellow ones available. Your myosin will, what's called bind, but you have the differences with myosin. It's actually, it has a key factor for so the key factor for actin is calcium. The key factor for myosin, though, your thick filament, is 100% ATP. That's like the main boy of myosin. ATP. Without it, you'll get something later on we'll see, rigor mortis, and etc. So what happens is, the key role is that ATP determines if the myosin is attached or detached. So you'll have an ATP right here. Even though it doesn't show it's ATP, they just have the phosphate labeled outwards, the third phosphate. So ATP head will go up, bind to the actin, then through hydrolysis, the phosphate group will fall off. It'll have ADP on it. That will initiate the power stroke movement, or a cross bridge will form first of all, and then a power stroke, which is when it pulls it to the M line. Cool? Now I want you to know this. The only way to take off this ADP now is to put another ATP to kick it out. And you could just think of it as like someone or something is occupying a certain thing like a seat. Like you put a backpack there, it's your backpack, right? That's actually a really bad example. Let me give you a better one. It's like you bought bananas from the store and you put them in a bowl. But you didn't touch the banana, so like you put in the bowl and there's the bananas. It's binded to the thing. Now the bananas are binded and blah, 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 but you haven't eaten them in like ages and they turned brown and nasty. So you gotta put something else in the bowl to take the place of the banana. So you eat out the bananas and you put new bananas.
Yeah.
Are you dying, fam?
No, the way is to just eat the banana. I was thinking it's more.
About Donkey Kong.
No, do you know the Indiana Jones, the first movie, when he switches out?
That's even better. It's like the little gold mummy thing.
The totem with like the rock.
Yeah.
That's what it is. So it exchanges and switches.
That's actually better.
So yeah, so like you can like the piston underneath it, it's still like down. So that means the myosin head is still attached when you switch it, and then finally when the ATP attaches, then it detaches. You see what I mean? Sure. Let's see if that makes sense to the people in the recordings.
Oh yeah, the riveter just came in.
Hey,
what up, Rosie?
We're just on sliding filament theory. I've been sliding filament theory.
Lecture 2, sliding filament theory.
Okay. What?
So don't worry, I had the first lecture recorded, though it might be like kind of a fuck up because I went too fast a bit.
But that's okay because I could easily just stop it.
Exactly.
Rewind it.
That's true. Hopefully I covered it well. I gave some puns and whatnot. And there is one thing I will actually show. She already has it, but YouTube is an acronym that I made.
Okay. Nice.
But that's essentially it. And then so you have another ATP bind that'll remove the myosin head from this part, and that'll cause no more cross bridge, and it'll relax the muscle. Cool? Or the myofibril in general. So I'll wait until you get your stuff set up, and then we'll start up right here.
I appreciate her making this and all that stuff, but it is the worst drawn out animation I've ever seen.
You actually get the animation?
No, I don't. But I mean, like, you know what I mean? Like, it's just so poorly done. Plus, it's always better in video form instead of slide by slide.
True that.
Oh my gosh.
I should mention, I didn't mention right here, Nancy,
Mm-hmm.
I should mention right here that they're pulled at a 45-degree angle. I'm not sure if she's ever gonna ask that, but just have for reference that it's pulled at a 45-degree angle.
Oh, you never know with her.
What's pulled?
The binding right here is at a 45-degree angle.
Oh, okay.
I don't know if she mentioned it in her thingamajig, in her podcast, and that's creating the power stroke. I'm not 100% sure if she's ever gonna bring it up on the exam, but just like, what's the degree of this? You'd be like 45. I don't know how I know, but
I know.
It'll be put in the back.
cool.
Got it.
Yes.
So once myosin has actually binded and pulled, the actin, it swivels back to the center. So it's not shown right here, but like, do you see how it binds behind? Like, see how like the whole actin like twisted to allow the myosin to bind? What happens is after it pulls, the myosin itself will actually swivel or get, because it wants to get off, right? So it'll just swivel off, and then like ATP will bind to it, and it'll release a phosphate. But just know there's like a swivel action. Now it's like basically that's it. That's exactly all you have to know. That it needs ATP and ADP to bind.
Okay, hold on. Really dumb basic question.
Mm.
The myosin head pushes the actin towards the M line. It doesn't pull it, right?
No, it pulls.
It pulls?
It pulls. That's what she said, if I'm not mistaken. Yeah, to pull the actin towards the M line. Which makes sense, because here, let me show you that picture, not this one. Like one of the breakdown pictures. Here we go. I can't zoom in. But you have this head, and here's the M line.
Yeah.
If I was to push, I would be pushing it towards the Z disc.
So the myosin head is like this, and then it conforms like that, so it pulls it.
Well, you could think of it as like, it's like this, and then it's just like, it's flexing. It's like curling its own arm, and it's pulling it towards the M line.
Got it.
I guess look at the shape of it. It kind of looks like a spring, so it's gonna pull like the arms in.
I don't know if you had already been in LARC, but
I don't have
May had said like think of it as like these are like the heads, so they literally just pull. They just go like that.
Got it.
But they pull towards the M line.
Got it.
Yeah.
Yeah.
Dude, are you all three in LARC?
Yeah.
I just started today.
I signed up last week. Yeah.
Your boy's like the only one not in it.
Yeah. It's actually really nice. Just
I love it.
Oh, but it
She's
costs
awesome.
money. And
yeah,
stuff.
but you're also paying for tuition here. $110 won't really make a difference in the long run.
It's not
I
money.
know it is. But
Well, I mean, I'm sharing all the docs with you anyway, so.
exactly.
That's
But
true.
then just having May in general.
Hey, nice.
Yeah.
Praise. All right.
Is that
but,
recording?
No. So, see, it even says right here. I'm sorry, I should have even pulled it right here. It says attempts to pull the actin towards the M.
Oh, I know. Okay.
And then once it's pulled it and it's swiveled back to the center, it'll release. It releases the ADP, ATP binds, etc. Because the new ATP molecule binds to the myosin, which breaks the actin-myosin bond. So as long as, let me put it to you this way. Where is it? Right here. As long as there's an ADP still attached to the myosin, it's binded to the actin. No matter what. You have to have ATP rebind to the myosin to make enough energy to break apart from the actin-myosin bond. Cool? And this is a key point, and I'm not sure if I'm going to be jumping a little bit, but think of it as like this in like a logical sense. For rigor mortis, for example, I died, right? And like your muscle will be fully, let's say I was like flexing somehow and I died, right? Since there's no more ATP being created, the muscle will be in a full contracted position or a tetanus type position. And since no ATP is formed, the bond won't break, right? Because there's no bond, or there's no more ATP being created, so the bond won't break, so it's still attached. But if I'm alive and kicking, ATP comes in, breaks that part, and then relaxes. Coolio? I can't take Nancy right now, bro. She looks so... She
looks beautiful?
No, not only that, but bro, she's like this. She's like, all right, I get this. Time to whoop some ass. Like, that's her look right now, bro. Like, all right, after this, we're going to run this steam engine, guys. Let's do this. Like, it's not a bad look. It's just like she's ready, like, geared up to go.
Okay, so ATP is bound.
So, yeah, and then ATP binder uses hydrolysis to break the bond. Cool? So, here's another. she says right here, once again, the heads are energized
But it's
and they can return to upright position. But it's just basically like the spring thing. So this is just a summary of it. She has like, I think, 4 points as a summary. Which I'll explain it a little bit more in the summary part. But it's basically here. So you contract, you're pulling in this. This is a quick question for Cass because she was the one here, but you guys can jump in. Cass, what is this part of the muscle called? It's the what compared to the trunk?
What?
She's getting a hang on. Remember the tree analogy?
The what? The branch?
No, the branch would be the distal part.
The leaf.
No, this would be your origin.
Oh.
Oh, that's what you're. Okay, got it. I was like tendon, muscle.
No.
Seriously, I
No,
was
no.
like extender, flexor.
Okay.
Origin is the one that doesn't have a lot of movement.
Yeah, it's just the one closest to the ball.
And then the insertion is the one that's actually.
It's your distal
so
one.
with your bicep, this would be the insertion. This would be the
So
origin.
right here,
Got it.
this middle chunk, that'd be your insertion part.
Yeah.
This is your origin,
Yeah.
and that's your trunk.
Yeah.
Cool.
Okay.
It was just a quick review. So here's the summary. So this is the part. So the main part right here is like you started. It's already binded, and you're ready in a flex state, and you already created power stroke. Cool. So you're already flexed. Everything got pulled. What happens is first ATP binds to the myosin, which causes hydrolysis to allow it to detach. Cool. Then ATP provides energy for the myosin to rotate and go back to its normal position. So think of it as like a two-part thing. ATP binds, breaks apart, and then there's still enough energy for it to return it back to its normal position. Cool? While it does that, the phosphate breaks off. So now it's just ADP, right? Another ATP comes due to the response of calcium, binding to it and causing the whole power stroke to happen again. And then just repeat. So this is what I was telling you about with rigor mortis. You have something called a rigor state. Now this happens naturally in us, which is just when like no ATP or ADP bind to the myosin, and it's just like very brief. I don't know how it's like, for example, I guess it's like sometimes when you wake up and it's like you got that like little numb tingly feeling. Cool. It's like, oh, it's not too bad. Nothing's binded to it. It's just like it's very brief.
Mm-hmm.
While rigor mortis is your dead boy. And then she went on like half a rant about stiff.
Seriously. My gosh.
Question.
Yeah.
What was the middle of the thingy again?
Sup?
What?
The middle of the
Oh,
muscle?
the
Insertion?
insert.
There you go. Thank you.
You tripped me. I was like, the middle what thing?
What thing? Myosin.
The myosin, the hinge, which part? So here's just a quick example of all that. So you have ATP bind to right here, right? See, you have an ADP right here. Cool? ATP comes in, it binds, breaks it. Makes it go back to its normal position. And you have ATP and P, right? Then you kick out the phosphate through hydrolysis, binds back to the globin or the G-actin, and then removes ADP, ATP comes in, etc., etc.
Wait, so doesn't ATP technically have a 3-part thing to it? Because ATP unbinds it from the actin, it brings it back to the cocked position, and then it also weakly binds it back to the actin.
What happens is right here you have ADP,
Yeah.
right?
Mm-hmm.
You have ATP come in and it causes the hydrolysis,
Yeah.
breaks the bond, and this goes back and everything relaxes, and this
Yeah.
goes back to its like starting position.
Yeah.
Cool. And then when it gets back to binding, like when it's like starting to rebind again, like you want to flex again, you still have the ATP binded there.
Yeah.
So what happens is you're going to have calcium come in, and that's going to make, or it's going to give a signal somewhat to the myosin to break off that phosphate, allowing it more energy to rebind back to the actin, and then it's bound to the actin, then you have ATP. You get what I meant? Or one more time.
One more time.
One more time.
Okay, so you have here. I'll do it on the thing. You have ADP right here.
Yeah.
Matter of fact, let's actually.
I wrote it in. It's fine.
Okay. You have ADP right here.
Yep.
Cool. And then ATP comes in,
Mm-hmm.
kicks out that ADP,
Mm-hmm.
right? Has enough energy because it's adenine triphosphate. Still has enough energy. Breaks the bond through hydrolysis.
Mm-hmm.
And then it returns this kiddo back to his normal position. Now, it might be like, oh, Ahmed, it's binded right here.
Yeah.
You have to remember that it's not bound at all until it gets the conformational change from calcium.
Okay, but in the last line of that number 2 box, the myosin binds weakly to actin.
I'm not sure about that because it's like an all or none. It'd be like weakly, like maybe it hasn't fully detached in some of the myosins. But you need calcium to give the conformational change for the troponin to move the tropomyosin to allow position for actin to allow myosin to bind to that position. But if you don't have any of the calcium in there, then it doesn't remove any of the conformational changes, so you can't really bind. That's what I'm confused about.
Got it.
So I'm presuming it's like an all or none movement.
Okay.
And then
From there I got it, yeah.
Oh, you're chill?
Yeah, after that I would go.
Chill? Chill? Okay. And this is just relaxation. So now when you want to relax, what happens is you want all that calcium to go away because calcium is still producing the conformational change, right? So what you do is now you're using calcium ATPase and you're pumping all that released calcium right back into the sarcoplasmic reticulum. Cool? And in this phase, you're also using ATP.
It's like you're releasing the prison, like the
Prisoners?
The
prisoners into like, what are they called?
The
Their
playpen.
cell?
Not their cell.
The field?
The
field.
holding? Oh.
Yeah, like you release them into the field and like call them all back to like their cells.
I guess.
Wait, so then when they're going into the SR, is that
That's
the
going
field
back-
or is that
that's
the cell?
going
No,
back
that's
to
going
the
back
cell.
into your cell.
Because
Okay.
it's in the cell, so they're going back into their sarcoplasmic reticulum.
There's a
cell.
riot, so you
Yeah.
need to use force to push them back into the cells.
That's true, because they don't want to go in.
Yay,
You're right.
prison.
You're right.
Yay,
Wow.
prison? Whoa!
Yay.
That turned really dark.
I know, I got worried. So here's just what I was saying. Calcium's pumped back then via the ATPase.
Okay, this is when I get lost.
The timing of EC coupling.
Yeah, this is the exact moment.
So this is the part where, let me see what I said exactly in my notes because I don't want to give you the incorrect info. All right. Anyways, the time of the cup. Essentially, it's like what I wrote here is like it's like a telephone. Like you're playing the game telephone.
Okay.
Right? It's like I tell Izzy something, then Izzy tells Nancy something, then Nancy tells Cass something. And all that, it takes time rather than if I just straight up told Cass what I wanted her to say.
Got it.
Cool? So that's basically what it's saying. You have a muscle twitch of a contraction or a relaxation cycle.
Yep.
Cool? And then there's a time or a short delay for all of what we talked about, all the signal to reach DHP, to hit up RYR, to release the calcium in the SR, all of that, even though it happens on the scale of like femtoseconds or picoseconds or whatever,
Nice.
all of that takes time, right? So you can think of it as even scale wise, it's like a minor time lag,
Got it.
right? And all of that time lag, in that whole time, you have nothing. Like there's like no net movement,
Got it.
right? And that's basically what it is. It's the timing of the EC coupling. And then it's like all that time until you release calcium and then it binds to the troponin. But before all that time lag, so the time lag starts from right here. All the
With
time.
the action potential.
With the action potential, because you get a signal from your brain.
Yeah.
Boom. Time lag for
AC,
all of this stuff to open. All of this stuff to depol. All of that signal to reach into the T tubule. To hit your DHP. To hit your RYR. Then finally releases your calcium. And then you get something occurring. But in between the whole time, in the span of like very, very small seconds, there's like a little lag.
So is EC coupling, that's from the action potential to the muscle movement or action potential to the release of calcium?
I'm guess it's time required for calcium release.
Okay, so it's action potential to calcium. Okay, got it. That's all I wanted. Thank you.
And that's what I meant by it's like telephone.
Got it.
Because it takes a little bit of time to reach.
So that's just what this graph is talking about?
So actually, this is a perfect graph. So right here you have...
Sorry, I hated this.
No, you're fine. So right here you have the motor end plate. I shot a signal, which took... I can't really see it right here. Time, what does it say? It shows no time frame. But here's the signal. Then by the time it reaches right around where the signal is traveling down towards the T-tubule, it took 2 seconds. Roughly. This is just... It says 2 seconds, I believe. Right? I can't see really. Someone can confirm.
2
Millisecond.
I'm sorry, 2 milliseconds. Then from right there, when it travels and hits, like the actual RYR or DHP RYR, that's when you have another big gap of it just releasing all of that calcium everywhere.
Got it.
Cool? And you have to remember, we're talking like all of this stuff, we're talking like, when we're always like using these examples, we're talking about one individual T-tubule. But if I go back to, where's that beautiful image? Oh, there we go. Past it. Oh, God. Oh, here we go. We have a shit ton of T-tubules that we have to all get on board. And it's like you have a bunch of rowdy kids. It's going to take a little bit of time to get that signal and all the calcium released evenly around.
Got it.
Right? Because you don't want to, like, I'm talking right now, but I don't want, like, part of my face to, like, not work while the other half is, like, working.
Yeah.
Got it.
It's like that sort of thing.
Okay.
Oops. Too far. So that's basically it for EC coupling.
So EC coupling is basically just the lag time it takes for the active potential to.
You just have a small little lag time.
Okay, that's it. Now I'm wondering if people with bad reflexes, what's happening in their brain?
That's what I was thinking right now.
Yeah.
Well, it's not that. If you remember from neuro, we also had like.
Absolutely not. I don't remember from neuro.
Oh.
Yeah.
Okay. Pause.
What do you mean?
Okay. Okay, so just for a reference, just a reminder, remember that we have certain parts of in our brains that are allocated to certain parts of our body.
Yeah.
Remember the human homunculus?
Yeah.
Oh, yeah.
And depending on how much information or how much power or resources you've allocated to a certain spot will like produce that sort of response faster or slower.
Got it. Okay.
Okay.
Stuff like, let's just say I was paralyzed from the legs down.
Mm-hmm.
And from a whole life I've never used them, and like some weird surgery came about, and it's like now I can use my legs.
Mm-hmm.
I have like, though I still have a small part of my cortex that's dedicated to legs. It's like very limited movement and whatnot, and I'll have to go through physical therapy, all that stuff to actually make that part bigger.
You would have the rewiring in your brain.
Yes.
Got it.
Yeah. So you have, I wrote it as 4. She doesn't mention the 4th one here, but I wrote it as 4. There are 4 things that require, so your muscle contractions always need ATP. I've shown you that through myosin, it always needs ATP to keep the cycle going, right? So you always need a constant stream of myosin. So you have 4 ways. She doesn't show the 4th one here, but she talked about it. So the first one is the phosphocreatine breakdown, and it's just a short burst.
Wait, 4 ways that ATP is produced?
Yes. There's 4 ways. I'll give you the 4th one like it's the last one. So you have phosphocreatine, which is like your burst of energy. Now you can think of this as like Nancy was walking and then she went into a room and then I jumped from behind the door and I was like, boo, and then she like bolted the fuck out, right? That was her using phosphocreatine. Her body, these are by the way in your muscles, phosphocreatine's in your muscle because it's like instant response. Phosphocreatine in your legs, instant response, and you just bolted.
So you basically end up using phosphocreatine for like the fight or flight.
For the fight or flight for like the first 10 seconds-ish. Right? There's other methods, which is when you have to use glucose, and these are rapid and energy efficient storage of it. So you have anaerobic, which you guys know, and aerobic. I'll just go over it again. Anaerobic is your quick boy. You don't have to use any oxygen at all, but not too much energy net. Right? Aerobic needs O2, so you need oxygen for it. It's slower, but you get a lot of energy released. So for example, for aerobic, it's like if you were running a marathon, right? You're breathing more, you need more oxygen, it'll produce more energy, but that's why like they do carb loading for that stuff too. Cool? And the 4th way, she doesn't mention it here, but it's something called beta oxidation. So I'll wait until you're good to go. Good to go?
Yeah.
Nancy?
Yeah. I know, so do I. I was like,
beta oxidation, now this one actually uses fat to create energy. It's the least one your body uses, and it's extremely slow. And for example, you would only use it in super like relaxed stuff. So you're sitting at home being a couch potato.
Always.
Your body would use beta oxidation. You fell asleep.
Always.
Or a nap. Or Nancy sleeps in class. JK, Izzy, mistake. You get beta oxidation activated, right?
I'm just using up all my energy. That's what's happening. I have too much energy. That's the problem.
You're using all the beta oxidation. But that's your slowest one. So speed-wise, it's slow for beta oxidation. Then comes aerobic, then anaerobic, and then phosphocreatine.
How much energy is produced from beta oxidation?
Very little, or it's extreme. Like you're sleeping, you're not using a lot of ATP when you're asleep.
Yeah.
So it doesn't produce that much.
Very little produced.
Otherwise, like if you fell asleep, you'd burn fat like super crazy.
Oh my gosh, is that how my body works? I wish. Okay.
But you have to understand phosphocreatine is actually, this is like a key thing, phosphocreatine is actually in your muscles. Okay? It's in your muscles, and you have something called creatine kinase that breaks it down. But you have to understand it's in your muscles. That's why it gives you that instant juice or the instant response.
Got it. And the other ones, like, actually, for the one for glucose, anaerobic and aerobic.
Yeah, those ones, like...
it's supplemented by something else, okay.
But, like, that one's in your muscle,
Alrighty.
It's limited time, and you just get a quick burst.
Got it.
Cool, yeah? So this is what I was telling you earlier, Cass, in the first lecture about the fatigue thing. That shitty fatigue joke. But anyways...
I can't wait to hear it.
No, it's too late. I'm not gonna say it again.
It's on the recording anyway.
That's true.
It's fine.
Anyways, so you have two forms of fatigue. Think of it as like when we say, oh my god, I have mental fatigue, and oh my god, I have physical fatigue. Cool. First one is central nervous system or central fatigue. That one's like very simple. You're just tired through like maybe you're like, I don't know. She said in the podcast, she just said it's down to like a lot of variables. But just think of it as like, easy. Nancy went to Disneyland. She like saw all the attractions and adventures and whatnot. She didn't like run around or anything, but you just saw like a bunch of lights and you saw a bunch of shows and you were introduced to a bunch of flavors of like food and whatnot. And you saw like music and singing. Then you come back home and your house is like all quiet and stuff, but your body's just like, I'm done. Like you can physically run like a mile or not, but your brain's just like, I'm done, bro. I'm out. Deuces.
Yeah, because what I ended up putting is it would be failure to generate an action potential. That's what I put.
Yeah.
So that's central fatigue?
Yes, for central. That's what I ended up putting.
I'll steal that.
Oh my gosh. Okay.
Good?
Yep.
Peripheral fatigue, which is just basically your muscle. So an easy way to remember this is central fatigue is like your core. Right? So that's like your brain and your spine. That's like the core sense of your being. Right? And your periphery is just like your outer part or your muscles that build up around the core.
Oh, it's just CNS versus PNS. Oh, duh. Okay, I got it.
And it's just your basic things. So when you're tired, for example, you, like, I'm going to say Cass, like, ran 20 miles. Her body will be physically fatigued when she goes back home and sleeps and wakes up the next day. She'll be physically fatigued. She's sore. Her body won't release a lot of calcium. Right? And due to that, she won't have, like, a lot of muscle movement. She'll have muscle movement, of course, but she won't have a lot of it. Because calcium is depleted. You can have hypokalemia through this. And you have a slowdown of your phosphate and calcium release. So by phosphate... that's like the phosphate part is part of the thick filament or your myosin, and then the calcium's for your thin filament, aka actin.
So you have low of each.
Low of each. That's why like if you like she ran the 10 miles and then she got down from her bed and then she's like starting to walk, her legs will be like, nope, fuck that, sit your ass down. We're not ready to move. Right? Sleep deprivation though screws up both your PNS and your CNS. So if you're sleep deprived, what's it called? You can have a very delayed reaction, and think about it as like this. You haven't slept in a while, and you're driving. And the person in front of you hits red light, your brain will try to react, but you're slow to react. You're like, oh my god, boom, my ionotropic receptors, let's do this. You remember? I'm like scarring you for life with my old stuff. That's an N110 or neuro thing, yeah. She was asleep at the wheel.
Honestly.
And then CNS, it'll be like I gave you a very easy problem, like let's say a math problem, like I gave you what's 50 times 50, and your brain will be like, oh, what? I don't know. Or I'll be like, what's 9 plus 10? And then I'll be like, what did it?
It took me a while. Shoot, I'm tired.
Or I told you what's 9 plus 10, and you're like 21. And I was like, come on, Nancy, it's 19.
That's the meme part of my brain working. Always working.
So you know what, man? It's week 5.
It's week 5.
Don't forget that sodium and potassium ATPase alters the membrane potential as well at this point.
Say it again.
Sodium, potassium, ATPase alters the membrane potential at this point.
Okay.
Cool?
Mm-hmm.
And then, I don't know why. Oh, here we go. I was like, why did I put relook at figure 12, 13? So the main reason why I said look at figure 12 for 13, and the main reason I do this for myself is mainly because she can legit, and I feel like she might do this, she'll ask us a question about one part that got fucked up here. Like she did with the tumor thing.
Yeah.
Right?
Oh, yeah.
So she might go let's see. So like all these 3 are peripheral, right? Your PNS, and these guys are CNS. But if I go, let's see, I changed something in your muscle membrane potential. That was like the last thing I told you about the sodium, potassium. They can change your membrane potential. You would have to know automatically, oh, that's got to be with my peripheral. Or, oh, that's got to be with my central.
Yeah.
So this might be a 1-point problem where she asked like A or B, like she did in the exam. Like, is it this or is it this? And you'd be like, oh, it's this.
Got it.
Cool.
Solid.
And they both can influence one another. Both your CNS can influence your PNS, and your PNS can influence your CNS.
Yeah.
Cool. So you have 4 types of muscle fibers, right? Each one has like a specific thing. So think of it as like us 4. Each one of us has something we're really good at. She's really good at dressing as Men in Black. Really good as Rosie the Riveter. She's good as Loki, and me as I don't want her to mention it because the jokes are too far. But each one has a specific, and I want to say this like in like broader term. Each one has a specific niche. Okay. Your slow type, or it's called ST type 1 or SO. Now this one is your endurance type. It has a lot of blood flow to it, and it's got low amount of force. That means it has very little myosin. Cool?
Wait, hold on. So lots of blood flow.
So it has a lot of blood flow. So this is like the one that has the highest endurance and a lot of blood flow, but it has low force. And by low force, it doesn't contract that much. So you can think of it as, since it doesn't contract that much, that means it doesn't have a lot of myosin. Because myosin is what produces the pulling force, sliding filament theory, etc., or the power stroke. Since it doesn't have that much force, that means it doesn't have that much myosin.
Got it.
Cool? Next one is you got fast twitch fibers, or FG, or fog type. And there's 2 of them. Even though she puts or, she's like sneaky as hell. There's actually 2 of them. There's type 2A, and then the other one is type 2X, right? Type 2X is your super fast boy, but type 2A is just your fast boy. Cool? So in this one, you have more force, and it's I'm going to say the type 2A first. So type 2A, you have more force than your type 1, but it's still not enough. You have some blood flow, but it produces a greater force than type 1, but not that much. Cool? Now I'm going to skip type 2X, or I'll just do type 2X. I wrote it out of order in my notes, but type 2X is your super fast one. Okay, this is like your fastest one. This one, it's like very limited, right? Very limited time, very limited power. It's super fast, and it doesn't require oxygen. But since it doesn't require oxygen, it's just like an instant like twitch. Like you gotta go fast, right? Like zoom zoom. That's your super fast one. So if she asks you which muscle fiber is the fastest, it's type 2X. And I think that one, she didn't mention it, but I'm pretty sure that one gets influenced the most by phosphocreatine because they're both act in a fast manner. It wouldn't make sense if phosphocreatine acted with like type 1, which is really slow.
Well, yeah, it makes sense because this is the very fastest one, and phosphocreatine produces the fastest.
Yes. And it doesn't require O2. That's why when you're when you're panicked and there's like parasympathetic and sympathetic's like when you see a line and you got a dip. That's like blood vessels dilate, everything blah. This one isn't affected by, it has blood, but it's not affected by O2, so you could just book it. Got it. But then you're going to have to rely later on your type 1 for sustained running. Cool? Your other one is called myoglobin receptors, or myoglobin. It's not really a muscle category or a muscle fiber type. It's just myoglobin, and it binds O2, and it's ready available for aerobic respiration. Right? And it's seen in fast processes because aerobic is like decently fast, right? If we go back upwards, where is it? Aerobic. Well, not here, not for the ATP production, but it's decently fast. It's like if I was like speed-wise, if I were to rank it, it'd be type 2X is the fastest, then type 2A, and then myoglobin, right? Mm-hmm. And then type 1. So myoglobin is important because, like, for example, depending on how much O2 is used by a certain muscle group, will actually change its properties. For example, since I know that type 2 or type 1A uses a lot of blood, that means it requires a lot of O2. Mm-hmm. So that means it'll have more myoglobin in it, right? That'll make the meat more white. She used that example like right here. More dark, because you use more oxygen. Mm-hmm. And she used the example of like a turkey, where you have dark meat and white meat in the turkey. The white meat, it's like your fast action, you gotta zoom, while your slow, which is your dark meat, which would be a lot of myoglobin in it, would be your slow boy, which would be darker. Cool? Now, and this is just showing you what I meant by fast twitch and slow twitch. So for example, you would have more type 2X in your calves, right? But you'd have more type 1A in your eyes, which makes sense because your eyes have a lot of blood to them, need a lot of focus. You're using them constantly to look around, pay attention, spatial awareness, etc. While your legs, you're walking with them, but when you're running from something, you're not running with your eyes or your hands, you're running with your legs, so you need like instant response. Mm-hmm. Cool? And like this is the structures level one, I guess, if you want to like look at your own time at the structures.
So I'm writing me over. Oh yeah, but that'll be independent reading.
So you can do biopsies, and then that's what she talked about.
To be able to see like the darkened up.
Yeah. Hang on, let me be like, all right, you guys can do this independently.
Yeah, you guys can do this independently.
Honestly, we have
all these tables. No, but basically all of this is just telling you about like type 2X, type 2A, and type 1.
Yeah.
So it just shows you like the amount of mitochondria, etc. So type 2X, low. Type 2, but it has like the lowest resistance to fatigue. So they instantly get tired and they're done. Mm-hmm. Anaerobic, because it's fast. And it's got a lot of ATPase, because if you need to produce a lot of ATP, you gotta have a lot of ATPase to break them down, right?
Yeah.
And I don't know what Vmax stands for, to be honest.
Velocity.
Yeah, velocity.
So it's
okay.
Speed.
The speed of the things all connecting and creating. Efficiency is very low, because you don't need to be very efficient to do something real quick, right? If we're playing, give me a sport.
Soccer.
If I'm playing soccer.
Football.
It works both ways because in Spanish it's the same.
Oh, whatever. I was talking about American, but alright.
Alright, okay. We'll just go with American. So, I can throw the ball as a QB anywhere. Doesn't matter. As I don't have to be efficient, I can just chuck a ball. That's like the instant response. But if I want to be more efficient, or like type 1, I'd be like, alright, I'm going to take my time, aim, I'm in the pocket, and then chuck it at someone specifically. And it's got very high specific tension. Cool? Type 2, which is what I consider your second fastest, or your intermediate, it has a decent amount of mitochondria. It's got a decent amount of resistance, and it's got about a combination of anaerobic and aerobic. It's not 50-50, but it's got a combination of them. Has a lot of, since it's fast as well, it has a lot of ATPase. Its velocity is medium, and it's got moderate efficiency, and it's somewhat high specific tension. Now, type 1A, which is your slowest boy, it's got high numbers of mitochondria because that's your endurance one. You need a lot of energy to keep your endurance ones kicking. That means it's got high resistance to fatigue. So your lactate, when you have a buildup of lactate, it's usually these two guys that get a lot of lactate buildup. That's why you feel more tired. These guys are like chill for a long time. You have a low amount of ATPase because it doesn't produce that much ATPase or ATP. It has slow because it doesn't have a lot of attachments to myosin. It doesn't have that much speed to it, right? Its efficiency is very high because it's like not using that much energy, and specific tension is moderate.
Why exactly specific tension?
I believe that's how like. I think that's talking about when it's talking about like titulin. Or Titan.
Oh,
I'm sorry.
Okay. Got it.
Thank
No,
you.
then I got it.
I was like, Tishulin?
I don't know where I think I've mixed Titan with Nebulin.
Oh, yeah, that's, I mean, it's basically the same.
Yeah.
But just in different places.
I think that's what it's talking about, like the specific spring tension of Titan.
Mm-hmm. Oh, which makes sense.
I'm not 100% sure. Cass has like that quiz, like, nah, that's not what it is, fam.
Mm-hmm. Okay.
Do you guys, did your Lark tell you guys something?
No.
No.
She didn't tell us anything about that.
Okay. We'll just go with that for now.
Mm-hmm.
And then this is just them doing an SDS page of it and like showing you what it is.
Yeah.
So this part is talking about the resting fiber length affects your tension on the muscle. She didn't actually go a lot on this one. She just like basically skipped it.
Yeah.
And she was like, hey, we're gonna jump to smooth, guys.
Was this the day that the fire alarm happened?
Yes, bam. But basically, your sarcomeres are contacted with an optimum amount of force. And I talked about this. That's like your optimum overlap. Remember that? Optimum overlap for you two that weren't here. It's basically when your myosins and actins are all fully. I didn't do that in the video, but like fully maxed and like stuck together.
Yeah.
Okay.
And you have the most cross bridges you could possibly have.
Mm-hmm. But it's
Cool.
not too much and not too little. It's still picture.
It's like your most
okay.
perfect.
Yeah.
Which, if you have too much of it, it could also cause tetanus, which is like 100% contraction.
Yeah.
Right? That's basically what it means here. She skipped it real quick, so I wasn't too sure if we had to know that. But she did quickly tag on one thing, which is something called motor neurons, which I will get into. Where did I write it? Oh, right here. So your motor neurons are specific to specific muscle fibers. So there's an image here I'll show you in just a minute. Right here. So you'll have like one motor neuron, and that one will go to a specific type of muscle fiber. They'll all go back to the central nervous system, but they're all connected to like one. So if I have like one muscle fiber, like I'm just saying muscle fiber A is the blue, B is the purple, and C is the red. I have one neuron connected to that, and then it has like a shit ton of branches that interconnect all of those. Cool? They intermingle with one another. So it's not like I can fire off, I mean, in a lab, you could probably fire off one. But like together, if I fire off motor unit 1, it'll fire off motor unit 2 and 3, right? It makes sense because it's like the most efficient way your body wants to be efficient, right? So 1 will be for type 2A, 1 will be for type 2X, and 1 will be for type 1. Cool? That's like each motor unit. And it's used that to better regulate the muscle. And then you're like, okay, so why does it connect to the spinal cord or that sort of thing? It's because you can actually use summation at this point. You can use a lot of signals to get better control of something. So it's like, what I thought about this is like when you just wake up in the morning, right? You have like very little control of your body. You swing your legs and you just like stand up to walk. You're not fully balanced yet sometimes, right? But then like as soon as you start walking towards the restroom, your body's like, oh, I gotta send off a lot of neurons, a lot of signals. I gotta get those guys focused. Cool? That's like about it, guys. Let me go to smooth muscle now. Oh, it's really chill. It's not that bad.
Wait, are we allowed to say this?
Is it
Oh,
4?
Yeah. 4 is it?
Until someone kicks us out, yeah.
Okay.
Old school rules apply.
Yeah.
Alright, so you have smooth muscle. Now, smooth muscle is, unlike skeletal muscle, smooth muscle is unicellular. Uninuclear.
Mononuclear. It's
Uninucleated.
Yeah, uninucleated, whatever.
Right? It's
One nucleus.
One nucleus. It's not striated at all.
Okay.
So remember, muscle is like in layers. This
More
one's not layered.
organization.
And the function of a smooth muscle depends on its location, right? So I could have like a smooth muscle, for example, in my intestine is different entirely than my urinary. I have one in my respiratory, 100% different from my ocular. Cool? You have two types of smooth muscle patterns, aka phasic and tonic, right? Which are very chill. I got you right here. So phasic is usually one that's relaxed. So you can think of this as like your throat. Your throat, if it ever wasn't relaxed, you'd be choking right now. But the time when it's not relaxed or it goes through its relaxed to contracted cycles, like you ate something, contracts to move the food down, slowly pushing it, blah, 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 right? But right now it's fully relaxed. That'd be your phasic ones, because it goes through a phase of relaxed and a phase of contraction. Cool? Your tonic smooth muscles are ones that are always contracted until they can relax, aka
your bladder.
Your bladder, yeah. You got no bars, girl. I was slowing down for
It's a bladder. Well,
I was gonna say sphincter, but you know.
Sphincter's
That too.
It's a bladder sphincter. That's what it is.
But anyways, so your bladder is fully always contracted.
So you don't pee everywhere.
Always contracted. It's true.
That's for the video. Yeah, you're welcome. For the visual.
If anybody ever watches this on YouTube that's not part of the class, I'll be like, yeah, she's
My
so
name
deep.
is. But yeah.
Anyways, so it's always contracted until you want to relax to release. Cool? Some smooth muscle actually, so this is what I meant by phasic. So phasic's like, oh, small time, like you swallowed something and then it goes back to relax, or it goes to relax to contracted, relax, contracted. While like, here's tonic, you're fully contracted, release, fully contracted, fully contracted. So this is another example of how smooth muscle coordination is different. So smooth muscle works as they all work together as in a unit, right? So they communicate through these ones right here, at least in your small intestine. They communicate through the gap junctions. Cool. So they're just like little it's like I had like right here. My notebook's next to her notepad, right? And it's like there's a little gap. That's what we communicate through. We're so stuck together, we communicate like that.
It's the nodes of Rambe is like similar to it. Almost.
Similar to that, man.
Yeah. That's like a visual. Someone
they work together, and they don't have like, depending on where they are, you can have some fine control or just like not like that much control. For example, in your intestines, you don't need that much control. While in your eye, they're more spaced out. They aren't connected by gap junctions, so you have more fine-tuned control with it. So your eye would work basically separately based on where it's depolarized.
Sorry, I
Yeah.
have an appointment for this room.
That's
Started
okay.
from 4.
Okay, yeah. Thank you.
Hmm?
Oh, let's stop the video.
Oh, that's probably a